Welcome to the Deep Dive, where we take a closer look at really interesting research publications. And today we're going to be looking at a paper from 2005, published in the Journal of Chromatography A. And the title of this paper is a mouthful: Viscous Fingering Induced Flow Instability in Multidimensional Liquid Chromatography. It's authored by, Kirsty J. Mayfield, R. Andrew Shalliker, Heather J. Catchpoole, Alan P. Sweeney, Victor Wong, and Georges Guiochon. Quite the team. Now, what we're really interested in today is understanding this concept of viscous fingering, the setup they used to study it, how they went about their investigation, and really what makes this research so significant.
Well, this paper really dives into this phenomenon, viscous fingering, and how it can really mess with the efficiency of multidimensional liquid chromatography, which, as you know, is an incredibly powerful separation technique used in so many different scientific fields.
Right. And for folks who may not be as familiar with this phenomenon, could you maybe give us a quick rundown of what viscous fingering is and why it presents such a challenge in chromatography?
Sure. So viscous fingering, often shortened to VF, happens when you have a less viscous fluid like water displacing a more viscous fluid like honey in a porous medium. Think about what happens if you pour water into a container of honey. The water, being less viscous, is going to kind of form these finger-like patterns as it tries to push into the thicker honey.
Oh, okay. I see. So how does this kind of uh, visual, I guess, translate to the world of chromatography at the very small scale?
So in chromatography, right, we're essentially separating different parts of a mixture as they move through this packed column. And this mixture is carried along by what we call the mobile phase, which is usually a solvent or a mix of solvents. So if there's a big difference in viscosity between what you're injecting, your sample, and the mobile phase carrying it, or even between different mobile phases, like you often see in multidimensional chromatography, you can get this viscous fingering happening. And this messes up those nice, neat bands of separated compounds that we're looking for, leading to poor resolution, basically making it hard to see and measure what's in there.
Yeah, it definitely sounds like a pretty big challenge, especially in multidimensional liquid chromatography, where you're working with multiple solvents and doing a lot of transferring between steps.
Yeah.
So how did these researchers go about investigating this phenomenon?
Well, they cleverly designed an experiment to kind of mimic the conditions you'd see in multidimensional liquid chromatography, but using a single column. They packed this column with non-porous silica particles. And by doing so, they basically eliminated any kind of broadening effects that might come from the stationary phase itself. And that allowed them to really hone in on the flow dynamics and see just the impact of viscous fingering.
Ah, so they isolated the variable, so to speak.
Precisely. And to actually visualize this whole process, they used a neat trick. They made the stationary phase inside the glass column invisible using a solvent that matched its refractive index. And this allowed them to clearly observe these fingers forming using a dye tracer that was injected into the flowing stream.
That's very clever. So they really went the extra mile to make this phenomenon visible. What were some of the specific solvent combinations that they tested, and what did they actually see happening?
Right. Well, they tested a few different combos, but one of the key findings came from injecting a large plug of acetonitrile into a flowing stream of water and methanol. That setup created a significant difference in viscosity, and they observed, pretty clear signs of viscous fingering. You could see it in the distorted peak shapes that showed up.
So the specific solvents they used and how viscous they were relative to each other really played a big role in this viscous fingering effect.
Yeah, exactly. And to make things even more interesting, they found that if they increase the amount of water in the mobile phase, which bumps up the viscosity contrast even more, the problem just got worse. You'd see even more distorted peak shapes. They also looked at how the volume they injected affected things. And as you might expect, larger volumes meant you got more pronounced viscous fingering.
Makes sense, right? Thinking back to that honey analogy, if you dump a whole bunch of water in, you're going to get those more dramatic and disruptive finger patterns.
That's a great way to picture it. And it highlights a really important challenge in multidimensional liquid chromatography, where you often have to transfer larger volumes between those separation steps.
Their experimental setup, the way they designed it, that sounds really innovative. It allowed them to see and study something that usually happens hidden away inside the column. Could you maybe walk us through the specifics of that setup? What instruments did they use? How did they put it all together?
Sure. So they used a pretty standard HPLC system with a UV detector. But the really clever part was the design of the column and how they used that refractive index matching. By choosing non-porous silica particles for the stationary phase, they essentially removed any contribution to the band broadening from the stationary phase itself. That meant that any peak distortion they saw had to be from the viscous fingering.
So they basically created a clean slate to isolate the viscous fingering effects.
Right. And the refractive index matching was key for actually seeing it happen. Because they matched the refractive index at the stationary phase with that of the solvent around it, the packed bed essentially disappeared. And that let them inject a dye tracer and watch those viscous fingering patterns forming in real time. They were even able to capture images, giving a visual proof of what was happening and how it affected the flow.
Being able to actually visualize it, that's pretty powerful. It's not just an abstract concept anymore. You have tangible evidence. So beyond just looking at it, did they use any other techniques to study the effects of this viscous fingering?
Yeah, absolutely. They very carefully analyzed the chromatograms that they got. They looked for peak broadening, where the peaks become wider and less defined, which basically means you're losing separation efficiency. And they also saw what we call peak fronting, where the front edge of the peak gets distorted, which again points to viscous fingering.
Okay, so they combined what they saw with hard data from the chromatograms to get a complete picture of what this viscous fingering phenomenon looks like.
Exactly. They documented all the changes in the peak shapes, measured things like peak width and asymmetry to quantify how much distortion was happening because of the viscous fingering. And they connected these findings back to the differences in viscosity between the solvents they used, really showing a strong link between the contrast in viscosity and how bad the viscous fingering got.
They were really thorough. It's clear that they've established a very strong connection between those viscosity differences and this viscous fingering in liquid chromatography.
Did their research explore any potential solutions or, ways to minimize this problem?
Yeah, they definitely did. Recognizing how important this is, they talked about some strategies to try to minimize viscous fingering in multidimensional chromatography. And one of their main recommendations was to be really careful about choosing your solvent combinations, making sure you minimize the viscosity differences between the mobile phases used in each step.
So it's about finding the right mix, getting a more balanced flow, so to speak. Precisely. And they really emphasize that even small differences in viscosity can make a big difference, especially when you're transferring larger volumes between separation stages, which happens a lot in multidimensional chromatography.
Understanding how all this works, the viscosity, the volume, how it all relates to viscous fingering, that seems crucial for really optimizing those separation methods. Do they offer any other, I guess, more practical ways to tackle this issue?
Yeah, they did. They suggested using something called a mixing chamber, basically a device that you put between the two separation dimensions. And what this chamber does is it ensures the mobile phases mix more gradually, smoothing out any abrupt viscosity changes that could trigger that viscous fingering. This leads to much better peak shapes.
It's like adding a buffer zone to smooth things out.
That's a good way to think about it. It's a relatively simple modification that can have a big impact. They also mentioned the possibility of tweaking the makeup of the mobile phase itself, maybe by adding a little bit of a more viscous solvent to a less viscous one. But they did caution that doing this can affect other things too, like selectivity and retention, so it's a bit of a balancing act.
Yeah, sounds like you have to find that sweet spot where you minimize the viscous fingering, but don't mess up the rest of the separation.
Absolutely. Their research really highlights how important it is to think about viscous fingering as a key factor when you're developing methods for multidimensional liquid chromatography. It's not just about the chemicals you're trying to separate, it's also about the physical properties of the solvents and how they affect the flow. You need to consider everything.
This is incredibly insightful. We've covered a lot of ground, from what viscous fingering is to how they actually made it visible and what can be done about it.
Mhm.
What stands out to you as the most significant takeaway from all this research?
I think the most important thing they did was raise awareness about the role of viscosity in multidimensional liquid chromatography, a role that's often overlooked. Their work has shown that if you don't consider and deal with viscous fingering, you could end up with some pretty significant errors in your analysis. And that's crucial for researchers in so many fields, whether they're working with pharmaceuticals, environmental samples, food chemistry, anything where accurate and reliable separations are essential.
And the way they visualize this phenomenon, that's something special too. It's amazing how they turned something invisible into something you can see and measure. Clearly, this research has really moved our understanding of multidimensional liquid chromatography forward. Now, before we delve further into the implications of their work, let's take a short break. When we come back, we'll talk about the broader impact of this research, what it means for the field of separation science as a whole.
What's great about this research is that it doesn't just point out the problem, it also offers some really good insights into how we can fix it. How to actually reduce viscous fingering in multidimensional chromatography.
Yeah, and that's the part that I think a lot of researchers are going to find really helpful. What are some of the big takeaways here? What advice would you give to chromatographers based on this work?
Well, I think the most important thing is to really think about the viscosity of the mobile phases you're using. I mean, it sounds simple, but it's something that often gets overlooked, especially when you're focusing on all these other separation parameters. But this research by Mayfield, Shalliker, Catchpoole, Sweeney, Wong, and Guiochon, they really drove home the point that even a small difference in viscosity between those mobile phases can have a huge impact on peak shape and resolution, especially when you're working with larger volumes.
So, in a practical sense, what can chromatographers do to minimize that risk of viscous fingering?
The most straightforward thing is to just choose solvent combinations where the viscosities are as close as possible, especially between the two dimensions of the separation. That means you need to have a good understanding of how different solvents behave, how their properties affect viscosity. For example, in a reverse phase system, if you keep the ratio of water to organic solvent pretty similar in both dimensions, that can help a lot. It reduces the contrast and makes for a smoother transition between the two environments.
It's almost like you're trying to fine tune the flow, make sure everything's interacting harmoniously, right?
Exactly. And something else they pointed out in the paper was the impact of the separation sequence, the order you do things in. They found that with one particular solvent combination, just by flipping the order of the separations, they saw a dramatic improvement in resolution. So that's something to think about too, especially how those viscosities are changing throughout the whole process.
Sometimes the simplest changes can make a big difference.
Absolutely. Now, I also talked about using a mixing chamber, which is basically a device you put between the two dimensions to make sure those mobile phases mix more gradually. It acts as a buffer zone, prevents those sudden changes in viscosity that can trigger the fingering. Pretty simple solution that can lead to much better peak shapes.
It's a clever idea, that buffer zone concept.
Yeah.
Do they have any other suggestions for mitigating viscous fingering?
Yeah, they mentioned adjusting the mobile phase composition itself, like adding a bit of a more viscous solvent to one that's less viscous. It's kind of like tweaking the proportions rather than switching solvents completely.
Yeah, like adding a little bit of thickness to even things out.
Right. But it's important to remember that when you mess with the mobile phase composition, you can also change other things like selectivity and retention. So you got to be careful, make sure you're not fixing one problem and creating another. It's all about balance.
Now, this research was mostly focused on those analytical scale separations, right? Do these findings translate to the preparative scale where you're working with larger quantities?
That's a good question. And it's definitely something to consider. While the basic principles of viscous fingering are the same, when you scale things up, the challenges and the solutions can be quite different. Remember, in preparative chromatography, you're often trying to isolate and purify compounds, not just analyze them. So purity and yield become really important.
So getting a pure product and getting enough of it, that's the main focus.
Exactly. And viscous fingering can really mess with both of those things. If you've got viscous fingering happening, it can distort those peaks, making it hard to collect clean fractions of what you're after. You might end up with a contaminated product, or you might lose a lot of your yield, which can be a big problem, especially on an industrial scale.
The cost implications are much higher when you're dealing with those larger quantities, right?
For sure. So preventing viscous fingering becomes even more crucial at that larger scale. Some of the things we talked about, like choosing solvents with similar viscosities and using mixing chambers, those are still good strategies, but you might need to adapt them for that larger scale. For example, a mixing chamber for prep chromatography is going to be designed differently than one used for analytical work because of the volume and flow rate differences.
It's interesting how scaling things up introduces this whole new set of considerations.
It does. And there are other techniques specific to prep chromatography that can help with viscous fingering. One common one is optimizing the packing material in the column. The material you use can have a big impact on how the liquid flows through the column and how likely you are to get viscous fingering.
It's not just what you're putting through the column, it's also the column itself.
Exactly. Particle size and pore structure, those are the key things to think about when choosing your packing material. Smaller particles generally give you better resolution, but they can also create more back pressure, which can be a problem when you're working with larger volumes. And then larger pores can handle more material, but you might end up with broader peaks. So it's a trade-off, finding that balance.
You have to carefully choose the right packing material to fit the job, right?
Yeah, it's all about optimization. Now, aside from the packing material, adjusting the flow rate and how you run your gradient can also help with viscous fingering. Slower flow rates can sometimes reduce the problem, but it also means your separation takes longer, and time is money in a lot of these industrial settings. And then your gradient profile is really important. A good gradient can help minimize those viscosity differences throughout the run, which reduces the chances of viscous fingering.
It's like you're conducting an orchestra, making sure all these different parts are working together in harmony, the flow rate, the gradient, the packing material.
I like that analogy. It really highlights how complex PRET chromatography can be compared to analytical separations. The stakes are higher, the volumes are bigger, and it takes a lot more fine-tuning to get things right. Now, there was one interesting observation they made in the paper about where the viscous fingering actually happens. In their setup, they saw it happening at the front edge of the band, which is kind of the opposite of what you usually see in prep chromatography.
Oh, really? That's interesting. Why would that be?
Well, it turns out that where the viscous fingering occurs depends on which fluid is more viscous. Remember, in their experiment, that injected plug of solvent, which was kind of mimicking that heart cut in multidimensional chromatography, it was less viscous than the mobile phase. So you get viscous fingering at the front because the less viscous fluid is pushing into the more viscous one. But in prep chromatography, it's often the other way around. The sample is usually more viscous, so the fingering happens at the back.
So it depends on the direction of that viscosity contrast.
Exactly. And this can actually be a helpful clue if you're trying to troubleshoot a separation. If you're seeing viscous fingering at the back, it might point to a different set of problems than if you're seeing it at the front.
It's like a detective using a clue to understand what happened. They also mentioned other factors, besides viscosity that could be affecting things like solvation effects.
Right. They acknowledge that some of the band distortions they were seeing might not be entirely from viscous fingering. Solvation effects, that's how the solute molecules interact with the solvent molecules. That can play a role too. Different solvents are better at dissolving certain molecules than others, so that affects how they behave during separation. If a compound isn't very soluble in the mobile phase, it might stick more to the stationary phase, and that can lead to things like broadening and distortion, similar to what you see with viscous fingering.
So it can be tricky to tell those two things apart, the viscous fingering and the solvation effects. You really need to have a good understanding of everything that's going on.
For sure, it's a complex system. And the researchers admitted that more work needs to be done to fully understand how these things interact, how viscous fingering and solvation effects work together to influence the separation.
Sounds like this research, while answering some questions, has also raised a bunch of new ones.
And that's the great thing about science. It's always pushing us to explore further. So, they've given us some great insights, but there's still a lot to learn. Like, why didn't they see viscous fingering when they injected methanol into a water methanol mixture, even though there was a viscosity difference? That's a bit of a head scratcher, and it suggests there might be something else going on, maybe some specific interactions between the methanol and the water. It's a reminder that we can't just look at numbers, we also need to think about the chemistry involved.
It's more than just physics. There's chemistry at play, too.
Absolutely. And they also said more research is needed to figure out how column diameter affects those viscous fingering patterns. The size of the column could influence the flow dynamics, especially at that larger preparative scale where you're using bigger columns.
It's like thinking about how the size of a pipe affects how water flows through it.
Yeah, good analogy. And then there's the whole question of how viscous fingering interacts with all the other stuff that's happening during a separation,
Yeah.
like diffusion and mass transfer. That's a whole area that needs to be explored further.
It's a complex system with lots of moving parts.
Right. And, viscous fingering, it might seem like a nuisance, something we just want to get rid of, but I think it's also an opportunity. It can teach us more about fluid dynamics, about separation science in general, about all these intricate factors that influence these processes.
It's not just about solving the problem. It's about deepening our understanding.
Yeah,
right.
Absolutely. And who knows, maybe someday, once we really understand viscous fingering, we might even be able to use it to our advantage. What if we could control those fingering patterns? use them to actually improve selectivity or peak shape. It's a bit of a wild idea, but it just shows you how much potential there is in this field for creative thinking. So yeah, this research by Mayfield, Shalliker, Catchpoole, Sweeney, Wong, and Guiochon, it's not only helped us understand this challenge in multidimensional liquid chromatography, it's also opened the door to a more complete understanding of separation science and all the possibilities for innovation that are out there.
It is fascinating to think about how we might be able to turn this problem into a solution, use our knowledge of viscous fingering to actually make our separations better. This deep dive into viscous fingering has been really eye-opening. We've talked about what it is, how it affects separations, what we can do about it, and the bigger picture of what it means for chromatography. So before we wrap up, let's take a moment to consider the lasting impact of this research, what it means for the field of separation science in the long run. Welcome back to the deep dive. We've been exploring viscous fingering and multidimensional liquid chromatography, really getting into the nitty-gritty of this phenomenon and what it means for separations.
It's been quite a journey, hasn't it? Starting from this seemingly simple idea and seeing how it can really have a profound impact on these sophisticated separation techniques.
Absolutely. And, as we wrap things up, I wanted to take a step back and think about the bigger picture. What lasting impact has this research had on the field of separation science?
Well, there's no doubt this research by Mayfield, Shalliker, Catchpoole, Sweeney, Wong, and Guiochon has left its mark. One of the most important things it did was to highlight just how complex these seemingly simple processes can be. Chromatography, it's used everywhere, but it's often seen as pretty straightforward. This research reminds us that at the microscopic level, there's a whole lot going on, all these intricate factors influencing how the separation happens.
It's like there's a hidden world, right? All these molecular interactions and flow dynamics that we need to understand if we want to get those really good separations.
Exactly. And understanding those underlying dynamics, that's key to developing more robust and reliable separation methods. And the paper really drives home the point that viscosity is a key parameter in multidimensional liquid chromatography, something you have to pay attention to. It's often overlooked, but they showed very clearly that even small differences in viscosity can have a big impact on the peak shapes and resolution.
It's a good reminder for chromatographers that you can't just focus on the obvious stuff. There are all these other factors at play that can affect how well your separation works.
For sure. This research really emphasizes that you need a holistic approach when developing your methods. You have to consider all these parameters, the viscosity, the solvent properties, column dimensions, even the order of your separations. It's like a multi-dimensional puzzle, and this research has given us some important pieces to work with.
And, another thing that really stood out was the way they used visualization techniques. The fact that they could actually see those viscous fingering patterns forming using those glass columns and the refractive index-matched solvents.
That was really innovative. It provided that undeniable proof that viscous fingering is a real phenomenon. It's not just an artifact or something. Seeing is believing, right? And those visualization techniques, they've opened up new ways to study and understand how things flow in these systems. And that's not just about solving the viscous fingering problem, that's a contribution to the whole field.
Yeah, they didn't just illuminate the problem, they gave us tools to address it and to keep exploring. And they also acknowledged that there's still more to learn, things like how specific solvent interactions come into play, how the column diameter matters, and how viscous fingering interacts with other phenomena.
Exactly. They answered some questions, but they also raised new ones, which is what good research does. It keeps us moving forward.
So where do we go from here? What's the future of research in viscous fingering and its impact on separations?
Well, I think we'll see a lot more work focused on developing strategies to mitigate viscous fingering in multi-dimensional liquid chromatography, especially at the preparative scale. That's where it really matters when you're trying to get those pure compounds in good yield. And I think we'll see more research looking at how viscous fingering interacts with other things, the solvation effects, temperature gradients, the challenges of separating complex mixtures.
So we need to dig deeper into the nuances, see how it all fits together.
Right. And maybe, just maybe, if we understand it well enough, we might even be able to use viscous fingering to our advantage. Imagine controlling those patterns to make our separations better, to get even sharper peaks. It's a wild thought, but it shows you the power of understanding these fundamental mechanisms.
It's amazing to think that something that starts as a problem could actually become a tool to improve our techniques.
Well, I think this deep dive into viscous fingering has given us a lot to think about. We've explored what it is, how it affects our separations, what we can do to minimize it, and its broader significance for chromatography. It's been a fascinating exploration of a phenomenon that often goes unseen, but has a big impact on what we do.
I agree. It's a great example of how science is full of surprises, even in something as seemingly routine as chromatography.
Well, thank you for joining us on this deep dive. We hope it sparked your curiosity about the world of separation science and the ongoing efforts to understand and refine these techniques that allow us to explore the composition of matter, to really understand what things are made of. It's been a pleasure. And for all of you listening out there, remember, whether you're a scientist or just someone who's curious about the world, there's always something new to discover. So keep exploring, keep asking questions, and keep diving deep into the fascinating world around us.